Laboratory in Bushel, Massachusetts. She did her PhD with Dr. David Caron on the phylogeny of Lactonin sarcodines in Woodley MIT undergraduate program in Bushel. She then moved to the Marine Biological Laboratory for a postdoc with Dr. Josephine on eukaryotic diversity. She is now an associate scientist in the Bay Power Center of the MBL and also holds an associate professorship at Brown University, where she also received her undergraduate degree. She works in stream and marine environments, focusing on how to explain patterns of microbial diversity and distribution over space and time, to understand how microbial communities are formed, and to reveal the role they play in ecosystem functioning. She was also the program manager for the International Census of Marine Microbes. Her research spans the fields of microbial ecology, molecular ecology, molecular evolution, cell physiology, phylogenetics, comparative molecular biology, and biogeography. She has more than 50 publications in these areas, so it is an honor for us to have the opportunity to learn more about her research activity.
Thank you very much. Can everyone hear me okay? Okay, thank you very much. I also want to extend thanks to the organizers of the ECODIM to invite me here to speak today, especially at a symposium in honor of Carl Woese. Today what I'm going to do is talk about my work as part of the Census of Marine Microbes that happened from 2004 until 2010. But I'm going to also look to the future in a program that's called Life in the Changing Ocean, which hopes to build on the successes of the Census of Marine Life. So this is a workshop and a symposium dedicated to Carl Woese, and it is really an honor to be here in his honor. And the reason for that is because Carl has played a big role in my career development. He mentored two of my graduate mentors. Ed DeLong taught me how to sequence DNA, and Mitch Sogan introduced me to the tree of life, and also taught me to be very critical of DNA sequences. And I want to mention a quote that I found on the internet that Carl made that hopefully drives home the importance of what I'm going to be talking about today in my lecture. And that is, he said, "It's clear to me that if you wiped all multicellular life forms off the face of the earth, microbial life might shift a tiny bit, but if microbial life were to disappear, that would be it. Instant death for the planet." So, we live on planet Earth, but perhaps we should be calling it planet Ocean. And that's because, as we all know, as part of the course, but also in general, that 71% of the planet is salt water. The average depth of the oceans is about 3,800 meters. And so 99% of the habitable volume is actually aquatic. Not necessarily by humans, but again, most of life is microbial, so most of the habitable volume is actually marine. But as sure as many students learned so far in the course, the oceans are hard to study, and so we know still not enough about them as we should. From an economic perspective, many countries actually have more ocean than land associated with their economic development. And this is including, of course, issues such as fishing and mining and energy. And Americans alone, I don't have a world estimate, but Americans alone consume 90 million metric tons of marine protein a year. So not only do we rely on the ocean for other types of resources, but also food. And finally, the top 2 feet of the ocean contains as much heat as the entire atmosphere. So the ocean is huge in terms of thinking about climate change. So I want to turn to the Census of Marine Life, and hopefully some of you at least have heard of the census. And this was a global engagement of scientists interested in better understanding the diversity, distribution, and abundance of marine life in the ocean, both past, present, and future. And specifically, what the census tried to do was to define the known, the unknown, and the unknowable with respect to marine diversity, specifically in the forms of species. It actually started as a program that was first trying to census fishes, and it began in the year 2000. There were grand challenge questions that came with the census, and these include the past, what did live in the oceans, the present, what does live in the oceans, and finally what will live in the oceans. And within here were nested 14 different what are called ocean realm projects. And all of these programs fed into what's called the Ocean Biogeographic Information System. And even though the census ended in 2010, this, what's now called IOGA, still lives on and provides a resource to the community to deposit data from census-like activities. So what were the benefits of the census? Clearly, I can't talk about all of the successes of the census, but what I do want to emphasize is that you need census-like data to be able to answer and address real issues in society, including things like sustainable fisheries, identification of marine protected areas and diversity hotspots, management of habitat loss and pollution, environmental assessments, identification and tracking of invasive species and also endangered species, understanding global climate change impacts, including ocean acidification, and finally the preservation of biodiversity and ecosystem services. So the census was a huge global undertaking. There were over 80 countries that were involved. It was spearheaded by the Alfred P. Sloan Foundation, which is a U.S. organization, but really that entity did not fund the science, it funded the research coordination. So that money was leveraged across 500 different institutions to be able to raise the money for the science that ultimately led to the data that was generated during the census. It engaged 2,700 scientists, including all the major continents, and really tried to be as encompassing as possible. Now, the benefit of the census data, of course, is that provided a baseline against which future change could be measured, and that's really critical for conservation-type efforts. So one of the legacies of the Census of Marine Life is the technology that was developed during the census. This is just a cartoon that shows all the different types of technologies, underwater vehicles, different types of remote sensing, tag-related data that was able to track marine organisms, and also genetic types of techniques that were used for the first time in many cases in many different parts of the ocean, which really helped to allow us to develop the kinds of data that ultimately ended up resulting as part of census efforts. In addition, the Census of Marine Life encompassed the entire ocean. So everywhere, these are some of the realms that were covered, everywhere from human edges, the near shore, coral reefs, the deep ocean, abyssal plains, vents, seamounts, the ice ocean, polar regions, and including the open ocean and the deep margins. All of this was covered. And my personal involvement was as part of the microbial ocean, which is shown here. And as everyone knows, the microbial ocean encompasses everything. So this was really quite a tall order. But in 2004, the organizers at in the census realized that they were missing a critical component. So even though this International Census of Marine Microbes didn't start in 2000, at least by 2004, the program realized that we really needed to study the microbes. And the sort of overarching goal of the International Census of Marine Microbes was to determine the range of genetic diversity and relative abundance of different microbial organisms at sampling sites throughout the world's oceans, specifically in an environmental context. And of course, we celebrated the 3 domains of life, the bacteria, the archaea, and the microbial eukaryotes, as part of this effort. Now, again, it amazed me that it took so long, if you will, 4 years out of a 10-year program seems like a long time, to acknowledge the fact that microbes, in fact, should be counted as part of the census activities. And I'd like to list this top 10 reasons why microbes matter, and this really did help in terms of trying to advertise and sell why microbes are important to study, particularly microbial diversity. First of all, the microbial world dominated most of the Earth history, to the tune of 50 to 90% of the Earth history. And I think it's safe to say that despite what happens to us, microbes will live on the planet Earth. Again, microbes are the primary engines of our biosphere. They're responsible for the major biogeochemical cycles in the ocean. They represent up to 95% of the biomass in the ocean and 50% of the biomass on land. There's estimated to be 100 types of unique microbes that may be associated with a given animal. And that's, again, a very underexplored part of what we understand. Microbes are important in the food we eat, in the decomposition of waste that we generate, in natural products that are useful to our health and also well-being, and also as fuel sources. So microbes are really important. And if nothing else, the reason we should care about microbes, if you don't care about the the ocean region, you still need to care about microbes, is we have 10 times more microbial cells in our bodies than we do human cells. So back in 2004, Mitch Sogan, shown here, and Jan de Leeuw came together and decided to submit a proposal to the Sloan Foundation to start the International Census of Marine Microbes. And I have to credit them for being the leaders in this program. I served as the program manager, and I was accompanied by a scientific organizing committee and also a scientific advisory committee. You may recognize Carlos here in the audience, who's going to be talking later in the program. But the point being that it was a really international program, and one that required the assistance of a very talented and diverse group of mentors and advisors to sort of bring us forth into the program. I also want to give special mention to Victor Gallardo, who was really instrumental in facilitating the Latin American ICOM, which we had a branch of as well, but also just in generating support for the microbial census in general. This is a photo that was taken in London at the celebration, the Natural History Museum, at the celebration at the end of the census. And unfortunately, some of our members could not attend that. So, the census of marine microbes. The ocean is a vast place. Most of the diversity on Earth is microbial. And so this was really a huge challenge for us. And we knew that going into this, but of course we also recognized how important it was to be able to start to understand the diversity of microbes in the ocean. And so in addition to being the most diverse organisms on Earth, and also being very important, microbes are hugely, hugely abundant. And so this just added to the complexity of the problem. They're very diverse, they're very abundant. This is just a breakdown of the different relative abundances, the concentrations of different major groups of bacteria, and I have also viruses here for reference in the ocean. So this was really a big undertaking, and one that we weren't really sure how we would approach. And thankfully, Mitch Sogan and colleagues came up with a really clever solution to this challenge of trying to at least begin to understanding the diversity of microbes in the ocean. And this was through the development of a type of technique called tag sequencing, sometimes called amplicon sequencing. And this strategy takes advantage of the ribosomal RNA gene, which Norm talked extensively about, and specifically targeting a short variable region. This technique, when it was first developed, was sort of at the cutting edge of the 454 pyrosequencing technology. At that time, you could only have really short reads, so this was a really great region to target. And so the way the technology works is basically you sequence, target just this hypervariable region, and each one of these tags serves as a proxy for a microbe in a community. And with the help of many long and well-sequenced genes, you can use these tags and query them against a reference database. And this is really critical to this technique. And when you combine this with massively parallel sequencing, what you get is not only taxonomic information, but you also get relative abundances of those organisms. But I want to caution that you can't build phylogenetic trees with tiny short sequences, and we weren't trying to do that with this method. But nevertheless, it really provided a first window, a first look into the diversity of microbes in the marine environment. So the census of marine microbes effectively started with a new technology, a new tool, and what we were able to do is solicit, we were able to get funding through the WM Foundation to purchase a pyrosequencing machine, but then also some money to advertise, call for proposals from the community. And this is just a distribution of the different projects that responded to our call, all the different samples that were collected. These circles are a log base 10 of the number of data that resulted. You can see it's a pretty good distribution from all of the major ocean basins. The red circles and the green circles are 454 data. But what people perhaps don't know is that one of the things that ICOM also did was collect legacy data. So we went back into the literature and we tried to organize legacy data in the in the literature to better understand some of the older distributions of diversity in microbial communities. And we also had a strong lipid component, which is something, again, a lot of people don't realize. This, of course, was the contribution from Jan Bril at the Royal Netherlands Institute of Sea Research, who made major contributions through his group to better understanding the lipid diversity within major groups of microbes. So just, again, to summarize, 1,200 sites, more than 25 million reads, and, again, international cooperation from 25 countries. So all of the data that came into the International Census of Marine Microbes. So I mentioned OBIS. This was the overall census database that provided services and storage of data for the Census of Marine Life. But one of the problems with the OBIS database is that at the time, no longer the case, but at the time, it only accepted sequences from named organisms. Now, as many of you know, or may not, there are about 10,000 or so described species of bacteria and archaea. Okay, so in the organisms that we were studying, clearly most of them had not been named. So this really was a big challenge for us. So we developed a new database called Microbes, which is where all of the data from the census were housed. Now you can access Microbes through a new website called vamps.mbl.eu. That VAMPS stands for Visual Analysis of Microbial Population Structures, and there's a portal through which you can access all of the microbial data from the census now. The data from the census, microbial census, included lipidomic data and mass spectrometry data, but also, of course, the vast amount of pyrotype sequencing and associated geospatial data. And finally, one of the perhaps richest components of the Microbes and the ICOM data was the highly contextualized metadata attached to that. So what is metadata? Metadata is information about the salinity and the temperature and nutrient concentrations at the time a particular sample was collected. Okay, so now getting to some of the findings of the International Census of Marine Microbes. One of the first papers, the first paper that unveiled the strategy that the census would use, the International Census of Marine Microbes would use, was a paper that was led by Mitch Sogan and colleagues, and led to the development and discovery of the rare biosphere, this concept of the rare biosphere. And I know that Carlos is going to be talking about this later on, so I'm not going to talk at it at length, but I just want to mention that this study, what it did is it targeted North Atlantic deep water samples and also samples from diffuse flows in the axial seamount that Julie Huber studied. And it looked at this sort of rank abundance curve, and what Mitch and colleagues discovered was that this pattern, this really intriguing pattern, has now been repeated in many other kinds of samples, wherein you have lots of low abundance populations and a relatively modest number of high abundance populations, and that a lot of the diversity, most of the diversity, is captured in the so-called rare biosphere. Now, Julie Huber went on to look very much more carefully at the axial seamount data and came up with some of the first estimates of richness, or numbers of species, within the archaea and also the bacteria, and was able to start thinking about numbers based on this clustering method that has been subsequently used by other groups, 3% and sometimes 6% clustering, but for the most part, the 3% clustering of the OTUs is what has sort of propagated mostly in the literature. And you'll see one of the important findings from this study was that archaea are several orders of magnitude lower in diversity than the bacteria. Another study which was really important, and I think sort of drove home the significance of the rare biosphere, is one that was led by John Barris's group and his student at the time, Billy Brazelton. And this particular study was one that focused on the Lost City hydrothermal vent, which is a really interesting environment, very high pH, very alkaline, and really interesting carbonate chemistry that involves a lot of methane, for example. And so these really established types of archaeal communities there, as well as bacterial, showed major shifts in the different ages of chimneys, carbonate chimneys, that the scientists studied. And what this showed is that there, over time, you have members of the rare biosphere blooming, if you will, and becoming abundant, depending on the ocean chemistry and the different environmental conditions and the age of the samples that they were studying. Now, as I mentioned at the start of my talk, the census was targeted at all 3 domains of microbial life, and this included microbial eukaryotes, not targeting the same region as bacteria and archaea, but a different region. And this study, led by Torsten Stoke, targeted anaerobic environments, specifically the Kariakor Basin and Framreven Basin in Norway. And this was a really nice study, because what it did is it unveiled major new phyla that were discovered as part of this census activity. So one of the goals of the census of marine life, of course, was to start thinking about the different numbers of taxa, of bacteria, archaea, and eukaryotes that are out there. And this is work that was led by one of the working groups that was in the final year of the International Census of Marine Microbes, called the Alpha Diversity Working Group. Alpha diversity is richness or number of species, okay? And so what this group, led by David Mark Welch, did was to try to estimate, this is the observed numbers of species in the entire ICOM data set, and then using techniques to estimate the richness as well. And as you'll see, there's several orders of magnitude difference between the estimated and observed numbers of bacteria and archaea. Within the bacteria, the proteobacteria were found to be the most speciose or richest. And within the proteobacteria, the delta proteobacteria, at least with the ICOM data set, were the richest as well. In addition to looking at just the total numbers, you can also start to think about different ocean realms, such as the epipelagic, this very surface, and then deeper and deeper into the depths of the ocean, as well as looking at vents or coral reefs. And you'll see this is the number of, total number of OTUs, as we call them, or species equivalents for bacteria. And you'll see that the richest parts of, at least the ICOM data set, included the bathyal, so the very deep waters, whereas places like the open ocean were much less speciose in terms of diversity. In addition, they studied bacterial richness varying by biomes, and these are what are called Longhurst zones, biogeographic zones of the ocean. And this is just to break down some of the more interesting biogeographic zones and how they vary. Again, some of the most some of the most richest areas are things sort of in this zone here, in these zones here, and some of the species poorer ones include things like the polar provinces. Okay, so the two previous slides that I showed you have actually never been formally published. But what's exciting about the ICOM data and the database that we've developed is that it's open to the public, so anyone can come and use the data. And in fact, there's a group, an independent group out of California, led by Joshua Ladau, who was able to use the ICOM data without any kind of assistance and develop models for richness in the global ocean. And so this is a data set that, again, the modeling used ICOM data and other data sets, but the ICOM data formed the basis of testing modeling on the bacterial richness in the global ocean. And what these scientists found was that in the wintertime, richness peaks in temperate and also high latitudes in the northern hemisphere, but in the southern hemisphere, temperate areas peaked in terms of richness in the summertime. So this is really exciting because one of the best things that can happen to any program is that not only do individuals get to publish their own data, but that you can use the data in a more synthetic way. And this is a beautiful example of that coming true, if you will. Now, in addition to alpha diversity or richness, we also looked at beta diversity, or comparing different samples in terms of composition of organisms. And this study, led by Zinger et al., looked at the differences between pelagic and benthic samples. I should add that not only did the census, the microbial census, target many of the major different oceans, but it also targeted the coastal ocean, the open ocean, the benthos, as well as the pelagic zone, and vents and microbial mats. So there was a broad diversity of types of environments as well, anoxic and oxic. So one of the main conclusions from this study is that there were huge differences between the pelagic and the benthic. I think they shared only about 10% of the community structure. And in addition, there were clear differences between the different types of communities, such as coastal, open ocean, deep ocean, etc. So that was an important synthetic product of the Census of Marine Microbes. From there have been other studies that have tried to test macroecological theory using microbial data. And that's another use of the microbial data that we've generated. This was a study that was led by a postdoc in my lab who looked at a classical distribution called the bipolar distribution. And the method that we used to do this work was one called null modeling. So it's a statistical approach, and we showed that there were actually fewer bipolar species than expected by chance using this null modeling approach. A similar type of approach was used by Jennifer Martini in her lab, led by her postdoc at the time, Anthony Amend. And one of the results, major findings from this work was that there was discovery that there are narrower ranges of bacteria than expected by chance using a null modeling technique. And more importantly, there was a trend in latitudinal ranges increasing with increasing latitude. And this was borne out in what's called the literal latitude range, the absolute latitude range. If you only looked at the southern hemisphere, you also found this pattern. If you only looked at northern hemisphere data, this was not a significant pattern. So it would be remiss to not mention some of the work that was done by the English Channel folks and Jack Gilbert. Jack was a huge proponent of the International Census of Marine Microbes. You may know Jack also from the Earth Microbiome Project, which was really inspired by ICOM. And this is a study that they did where they sequenced the English Channel samples to the depth of 10 million sequences. And what they found is when they compared their data across all of the different, well, 350 some odd samples from the census, they found that they could actually recover something like 30 to 60% of the whole ICOM data set. And he argued that this meant that there was this persistent seed bank throughout the ocean that is drawing upon, that can be drawn upon at any given time. And certainly within the English Channel, between different seasons, you could actually find the very rare taxa occurring throughout many different years of study. So I want to shift gears a little bit now and talk a little bit about my own work. I mentioned that Mitch Sogan was my mentor. He introduced me to the tree of life, and he also introduced me to thinking holistically about diversity on Earth. So I don't just work on bacteria, I don't just work on archaea, I work on all three domains of life. And a project that has facilitated that is one that was funded through the National Science Foundation. And it was called, my project was called Microbial Inventory Research Across Diverse Aquatic LTERs, or Mirada LTERs, which I like, because what it was a snapshot of the microbial diversity across the long-term ecological research sites that are funded through the National Science Foundation in the US, although some of the sites are international. And this program allowed me to look across very diverse environments such as open ocean and polar regions, as well as coastal and freshwater. So it wasn't just marine. There's some really interesting insights you can gain by looking across domains and also across different types of aquatic environments. Now, the strategy that I used was similar to the one that was used by the Census of Marine Microbes, in that we targeted the exact same technology, the V6 region for bacteria and archaea, but what my lab contributed was the introduction of using the V9 for eukaryotes. And this was actually a protocol that I developed to apply to ICOM as well as Mirada samples. The reason for choosing a different variable region for eukaryotes is simply that the V6 region in eukaryotes is not very variable. Another reason was that the V9 tends to be pretty standard in length for the most part in eukaryotes. It's also one of the extreme hypervariable regions as well. So I just want to share some of the results across the domains and some of the insights we might be able to gain from that. For example, this is bacterial alpha diversity or richness. These are all the different stations we've worked with, and estimated richness along this line. And I might add that we've used a parametric diversity estimate, which is supposed to be a better representation of the actual richness in a given sample. And what we've been able to do is compare and contrast richness across fresh and marine samples. And what we find is that for the most part, within the bacteria at least, the fresh water environments tend to be more diverse than marine ones. The coastal environments, more diverse than open ocean. And finally, brackish environments are really dependent on your definition of brackish and also where your sample came from. And I just want to add that coral reefs were indeed more diverse than other parts of the ocean, at least in this limited data set. When we look at archaea, we find a different pattern, interestingly enough. With archaea, freshness doesn't seem to have as much of an impact on archaeal diversity. However, coastal samples were once again more diverse than open ocean, and tropical weren't necessarily the most diverse. And again, a similar pattern with brackish stations that were very variable depending upon the given sample. And finally, microbial eukaryotic alpha diversity. We also examined, as with bacteria, freshwater were in general more diverse than, or at least the most diverse samples were within the fresh realm. But unlike bacteria and archaea, coastal and open ocean comparisons were more or less comparable. And in the case of coral reefs, there tended to be a higher diversity in some, but not all, coral reef samples. And finally, brackish stations, again, depended on the definition of brackish. So one of the things that has been clear and has already been published in the literature is that salinity definitely matters across domains, but the patterns differ. It's mostly been studied in eukaryotes. There's a curve that has been developed called the Raman curve, and what it shows is that diversity dips in brackish environments. And amazingly enough, we did see a very similar pattern in microbial eukaryotes. But within bacteria and archaea, there seems to be some differences in how these organisms, these domains of life, are responding. You can also look, leave alpha diversity and start to look at patterns of beta diversity. And as with bacteria, you really do see differences across the archaea and the eukaryotes as well. So clearly, salinity really does make a difference even within these other domains of life as well. So, as you can imagine, there's still many unanswered questions about marine microbes. We still are boggled by the fact that there are so many microbes out there. Also, there are many, many microbial niches we have not studied, and this is really important to think about in terms of the completeness of the census. We still don't know really what governs endemic and global distributions. There are people that would argue that perhaps there aren't, in fact, endemic species. And then finally, what is this role of the rare biosphere? And Carlos is going to be educating us on that later. Why do microbes matter? I've already mentioned this. Habitability, global climate change, ocean acidification, ecosystem services. These are huge issues, and microbes simply cannot be ignored in our understanding of them. So the solution, we hope, is something called life in a changing ocean. And the reason, unfortunately, I have to leave you early is because I'm going to be flying to Montreal immediately after this meeting to join colleagues in Canada who are starting to try to provide funding for a program called Life in a Changing Ocean, whose tagline, if you will, is biodiversity discovery and knowledge to support healthy and sustainable ocean ecosystems. The program has 3 major themes. And these include biodiversity discovery in space and time, biodiversity and ecosystems functions and services, the why, and finally biodiversity and human exploitation. So this sort of what we call follow-on census program is not intended to be a second census, but at the same time, all of the people that are involved really appreciate the fact that we simply have not discovered all the diversity that's out there. We don't want to abandon that. But we want to expand it to be able to think about functional diversity, and also this aspect of the human dimension of diversity and how we personally have impacted diversity. Now, it's a little embarrassing to show this slide because it really points to the fact that how poorly we've sampled the census as part of even ICOM, which, you know, I like to say was extensive, but it really wasn't. This is a picture of the different, this is the globe divided up into grids, and these are the grids that we sampled. There's clearly a lot of samples that we have not been able to, parts of the ocean we haven't been able to get to, things we haven't been able to sample. In fact, we estimated that we've sampled something like 1 divided by 10 to the 18th the volume of the ocean. So anyone that tells you they've sampled the ocean well, I don't believe them. So what I've been trying to do personally is to leverage ships of opportunity. This is a vessel called the Corwith Kramer. And there are different cruise tracks. This is a program in Woods Hole that is an educational program where they've been doing regular cruise tracks and provide a method of opportunity to collect additional samples. So there are other examples, many examples of these kinds of opportunities that we should be taking advantage of to try to continue some of the work that the census started. In particular, I mentioned niches that we haven't sampled. One of the ones that I've been involved with personally in my own research is a place in the ocean that's new. It's about 50 years old. I call it the plastisphere. It's a place where plastic from land and also from marine sources has accumulated in ocean gyres, and I've been looking at the microbial diversity attached to those communities. And what we find is they're really different than the seawater, the surrounding seawater. And so one of the interests I have is trying to better understand the impact of these communities on the open ocean, which is really a microbial desert, if you wish, in terms of diversity, but also in terms of nutrient concentration. So we're interested in how this plastosphere is changing the fundamental nature of the ocean. Also, I want to put in a plug for a new program that's part of a European program called MB3, which stands for Marine Microbial Biodiversity, Biotechnology, and Bioinformatics. Thank you. How could I forget the most important one? Anyway, it's called Ocean Sampling Day, and this is one of the working group packages that's part of this program. It's in addition to having standard protocols, it also has a uniform sampling event of June 21st, which is, of course, the summer solstice. And it's orchestrated. There will be some sequencing by the Earth Microbiome Project from Jack Gilbert, and also, again, very important contextualized data that will be drawn from the different samples. This is an open call. So, and one thing I didn't mention is there is right now really a limit of samples that have been collected in the southern hemisphere, and I think if there is interest, it would be great to see colleagues here in Concepción and elsewhere in South America contributing to this effort. So I want to put in a plug for that. Also, I mentioned the aspect of time. Both my project Mirada was a snapshot in time. In some cases, we sampled 2 different seasons, but really pretty minimal understanding of how things change over time. And this is really important. In 2010, I served on a steering committee where I was asked to advise on what the requirements or sort of the characteristics of a marine biodiversity observation network might look like. And our paper was recently published last year in Bioscience. And in that paper, we provide recommendations. And I'm happy to say that among them are the microbes, but also even more holistic is looking at all taxa at all levels. And we feel this is really important. And much to our pleasure, there has been a recent call in the US, headed by NOC, and most recently the European Union, to establish marine biodiversity observation networks. And I'm really excited about these opportunities because I think they're really where our science needs to be headed. Okay, so I am going to now acknowledge the folks that I've worked with at the LTERs. Of course, there are many, many people. These shown in red are ones, LTERs that were based at the MBL. I may not have mentioned this, there are 26 of these. I've worked with 13 of these, so this was, again, a pretty large coordination effort. Incidentally, this overlapped with the Census of Marine Microbes, so I had a lot of coordination under my belt during those years. But it was a really exciting project, and we're still publishing very much from these projects. And finally, I need to acknowledge all the people that were involved in the International Census of Marine Microbes. Again, we had an illustrious organizing committee and an advisory committee, which we're very indebted to. But we also had really a wonderful community of scientists that responded to our request for samples, and that hopefully have in turn benefited from that. The Keck Foundation and the Alfred P. Sloan Foundation funded this work. The NSF funded my own work. And this is just a list of all the collaborators that participated. So I will end with that, and thank you for your attention.
So thank you very much, Linda, for an interesting talk. We have some minutes for a few questions before the coffee break.
Everyone wants coffee. Has everyone heard of the census before? Yes. Yes.
I have a question.
Yes.
With the tag sequencing, how do you deal with what I'm just getting into it with Nicole and Rodrigo and Osvaldo at why-paid those that but
That's very important.
yeah, but one thing I don't get is the units of biodiversity that one's measuring, and the worry I have is that it might change from group to group. So, for example, in eukaryotic phytoplankton, it's very well known of some examples, I think also in bacteria, where you can have things that are identical at the 18S, but they have different distributions, that they look different under the microscope, they behave differently, and they occupy different, they're obviously different.
Yeah.
But the akinets are identical across the entire thing. And
Yeah.
so,
Well, you know, this is a problem for really any gene that has differential rate of evolution, right? So you're going to have some genes that are faster than others, and it's especially extreme in small to ribosomal RNAs. You know, the solution to that is probably to, you know, look at a different gene for a given group. But it is another strategy that you can use is one that is a strategy that's been developed by a postdoc at the MBL called oligotyping. And, you know, most people use that 3% cutoff, right? There's a lot of diversity that's lost in that 3%. That, you know, if you go up to 99% or even 100%, you know, cutoffs, you can start to pick apart some what are called ecotypes. And there's a technique called oligotyping that lets you start to pick apart sort of ecotype-like differentiations in groups of organisms. You know, you're always going to be constrained if you have identity. If you're exactly the same, it's going to be a problem. But the more data you have, you can actually start to see very fine details between different groups of organisms, even at the 99% level. And I think that's one solution to this issue that you're describing. But you know, no molecule is going to be perfect for every organism. The best you can do is to be able to tailor your metric to your question.
Yeah, I was wondering specifically actually in the case of the stuff you presented where the archaea appeared much less diverse than the bacteria.
Mm-hmm.
And I'm wondering is that a real, I don't know what we say as a species concept for but is that a real diversity difference, or is that because this marker evolves less fast in archaea than bacteria? And how close? I mean, we'll always have that problem, but how much do you have any idea in that case?
Yeah, I mean that's a great question. Clearly, it's very difficult to answer that question. But personally, I feel that if you look at metabolic diversity of archaea, it is definitely lower than bacteria. So if you want to use that gauge, comparative gauge, I think it is reasonable that archaea are less diverse. That's one way to look at it. Beyond that, you know, we just have to be able to, you know, be more expansive in our sampling and show the fact that despite, you know, having sampled all what we believe to be major niches out there in the world and the ocean, you know, we're still getting similar numbers. What's interesting about the archaea is it's pretty consistent between studies, so I don't think it's methodological. I really do think they're just less diverse. Do you differ? No.
I believe you.
Yeah. You know, but I think part of their, you know, their ability to adapt to different environments is more constrained, and I think that's probably part of it. I mean, that's just mine. Anyone else?
Linda, you were part of our course during the last 2 days, and you heard our elevator talks in the evening to some of them.
Yes.
But the word plastic sphere appeared to me as one of those catchwords.
Plastosphere, yes.
Could you elaborate? What do you want to do there?
What do I want to do there? Right. So, so the Plastosphere is a great project because one of the things it does is it engages students, in fact, on board the sailing school vessel that I showed you. I was going to talk about the Plastosphere more tomorrow because I've been invited to give a short lecture for you guys. So I will describe that more in length then. But effectively what the Plastosphere project is doing is it's collecting these microplastics that are out there in the ocean, and we're better trying to understand the diversity of the microplastics, basically the function, how they function in terms of a community, because we find, in fact, all 3 domains of life on the Plastosphere. And finally, the fate. What happens to plastic in the ocean? We know that it can travel very quickly to the ocean gyres, but does it bring with it its original community from the land? Or does it develop a new community as it moves through these ocean gyres? Those are the types of questions we're asking. With our first study, we actually discovered that there were a lot of Vibrio communities associated with these plastics. So one of the things we're really interested in is what kinds of Vibrios are they? Are they potentially harmful to humans or other organisms such as fishes? That's another question that we're asking. We're also really interested in how the plastosphere is changing the oligotrophic ocean, because the oligotrophic ocean is very bereft of nutrients, and plastics have a way of sucking up metals, which can be critical to the growth of organisms, particularly iron, in the open ocean. Also phosphate. There's many different properties within those spheres of plastic and the life associated with them that may in fact really change the way we look at the open ocean deserts, if you will. And so those are all questions that we're very interested in. And of course, we are interested in the potential for these microbes to degrade plastic, whether physically or metabolically.
Great. You didn't use your 2 minutes.
Oh. Did I miss any keywords?
No. Great.
Thank you. Okay. Anyone? Yes?
Norm commented on the importance of relying on observation and worrying about estimation. And a lot of the comparisons were estimated richness. And I wondered if you could comment on Bart Hageman and Joshua White's paper in his measurement, suggesting that richness estimators are worth nothing unless they're Simpsons or Shannon diversity.
Except if they're what?
The Hill indices, Simpsons and Shannon diversity.
Are worth nothing?
No, those
Oh,
are the
unless
good
they're
ones.
Hill and, okay.
Yeah.
Well,
Have you
okay,
read the
so
changes?
the methods I've been using are parametric estimates. They provide confidence bounds. So you can actually have some sort of certainty with which to bound your richness estimate. They're the best estimates that I'm aware of in the statistical, ecological statistical world that you can use. That's not to say you can't use other methods, and that is certainly something that we will do. But model-based methods are certainly, I think, considered a better step. If there are any richness estimators that you can use, those would be the ones that you should try to use. That's my philosophy. With eukaryotes, interestingly enough, a lot of people use abundance-based methods, which I do not agree with. And that's because I think someone raised the point earlier that copy number in eukaryotes goes anywhere between 1 to 10,000. So if you're trying to use relative abundance with that kind of a statistic, it becomes really tricky. So one of the things we've done, and I didn't mention this, I should have, the Mirada samples in particular were replicated samples. They were from 8 stations, sampled and replicated from each of the LTERs. And we used that replicated sample to do binary or presence-absence-based estimation. Again, somewhat model-based, and that, I think, is an approach that you can use to sort of get around this issue of having this differential copy number and having bias by abundance. The abundance problem is real, there's no question, but those are the tools you have in front of you effectively. I mean, ultimately, hopefully we'll be able to have probes for every microbe that's present and do real counting. That would be the future, I guess, looking into my crystal ball. But that's a little ways off, clearly. Someone else had their hand raised? No? Well, we can talk more in the...
Yeah, we can continue the discussion in the coffee break now. Thank you.
Que de mi jefecita. Vamos a invitar a este mismo para hacer la busca.